So good morning, everyone, and welcome to the third in the series of webinars from the Industrial Transformation Research Hub in Biocatalysis for Pharma. Before we start, I'll just mention that we've got a number of seminars that are coming up in the next few weeks, and the schedule for these is shown in the next slide. So today, as you can see, we've got not one, but two speakers, both of whom are from UQ, University of Queensland, Professor David Ascher and Dr. Lisette Pregelj. So just a bit about them before we start. So David trained as a biochemist and structural biologist, but has since moved into the computational area. And he has developed specialist expertise in deep or machine learning and AI, artificial intelligence, as applied to proteins and their interactions with ligands. Lisette, on the other hand, is an interdisciplinary researcher across innovation and biotechnology, having trained as a biotechnologist originally. And she has a specific interest in the speed of innovation and in new product development efficiency. So today they'll jointly tell you about evidence-based approaches that can be used to streamline decision-making for biocatalysis. So without further ado, I'll leave it to David to tell us about how he applies deep learning to the sorts of problems that are relevant to pharma and that we want to address in the hub. Thanks, David.
Thank you very much, Liz. And thank you everyone for taking time in your very early mornings to come listen to us today. In the hub, we're really focused on evidence-based decision-making to really guide what we're trying to do. And today we're going to cover this from really two strong aspects. One is we really want to be informed from industry, from the business side, about the sorts of problems we're working on and the solutions for those problems, but also then feeding back into how we can evaluate whether these solutions are actually cost effective and having the impact we want them to have. And so Lisette's going to talk to you a bit about that today. And then I'm going to talk a little bit about how we're thinking that we can apply the use of artificial intelligence and computational approaches to really streamline the full diversity of applications that we are trying to run, right from the business analysis through to how we can delve into and optimize rationally the sorts of enzymes and proteins and modifications to those that we really want to explore using a broad range of particularly artificial intelligence networks. And so focusing a little bit in on that deep learning and machine learning framework, one of the ideas that we've really come into, particularly in conversations with our industry partners, is how we can integrate some of the tools that we've been developing here at UQ to really guide the selection of enzymes for further optimization and help improve the optimization. So Liz and her team and Gary Schenk, who are very well versed in the application of experimental approaches to guide and explore broad ranges of chemical space and optimization of those enzymes, have generated a large amount of data that we can now use to train and optimize our AI algorithms to hopefully optimize the approaches in which we want to use to select which enzymes and which modified enzymes we want to explore. So you would have heard from Liz a couple of weeks ago about how she has really successfully been using ancestral evolutionary sequence reconstruction in order to try and design and find enzymes that are going to be more thermostable or catalytically active. But one of the issues with that is you'll often generate a very large number of potential sequences that you could explore, and we need ways to identify which ones are we really going to look at. And if you just think about, a simple 200, 400 amino acid enzyme, once you start thinking about all the different changes you can make to each amino acid residue across that full sequence, and then the permutations and combinations of those, the numbers expand exponentially, and we need ways to really explore this very broad enzyme space in a high throughput manner, and hopefully be able to do that in a much more efficient fashion. And so where we're proposing to do that is particularly through the application of AI algorithms. Now, a lot of you, it's not going to let me do, a lot of you will be familiar with AI and its application within broader biology. But generally, the way AI has been applied across bioinformatics has really relied upon very simplistic representations of the data. So at a protein level or a DNA level, most AI algorithms have really relied upon sequence information. And at a small molecule level, a lot of them use things like SMILES representations of those small molecules. From my background on more structural biology, I really had an appreciation for the complexity that three-dimensional structure can bring to these problems, and the fact that even though structural information might be encoded by that primary information, you often lose a lot of information by just considering that sequence space. And so we had the proposal about 10 years ago now that we really needed to integrate into our AI approaches more complex representations of the biological data, whether that be the three-dimensional structures of the proteins, the interactions they're making, or even more broader holistic systems biology type approaches. So capturing the broad networks of interactions that proteins and small molecules might be making. And so the first problem that we had when we sort of entered this sphere is that a lot of the approaches for being able to represent sequence-based information were really well described. And even if you think about even the more recent introductions of large language models, there's a lot of approaches for capturing sequence-based information in a large language model, but there's a lot fewer ways in which we can do that and capture more complex three-dimensional information. And so we really pioneered the development of two main ways to try and capture this three-dimensional information for the development of AI algorithms. The first of those that we developed, we termed graph-based signatures, and it was really a somewhat stolen concept from what computer scientists have been using to predict changes in weather or traffic. And we applied the same concept to how we thought we could represent a protein structure. And what we did was we labeled each atom of the protein as a node. So each node was an atom of the protein, and the distances between those atoms were our edges. And we've evolved this over the time, so we've now moved away from just counting the distances between atoms to actually looking at the types of interactions they are making. And so we can label our nodes with the types of the atoms. So, is it hydrophobic atom? Is it going to potentially make a hydrogen bond donor or a receptor? And then we can label the types of interactions that are being made to try and capture not just the chemistry of the protein, but also that geospatial arrangement that's going on. The other way that we've been really pushing into the space more recently is what was termed as protein structural language. And so what we're doing here is we've essentially taken the concept that, well, you're probably all familiar with the secondary structure elements of a protein, an alpha helix and a beta sheet and a loop. But each of those elements have different properties associated with them. And so what we can do is we can label each amino acid within the sequence based upon the properties of those secondary structural elements. So is it on an alpha helix? Is it exposed or a buried alpha helix? Is it a three-turn? Is it near the end? Is it on a single beta strand, or is it on an anti-parallel beta sheet? And we can capture that in a broad range of more complex languages. And so we can turn a protein sequence from 20 amino acids into about 240 different protein structural language characters, which can then be used to build our large language models or other AI algorithms. And so just focusing in on that second approach a little bit more closely, this is this sort of the broader representation of that we're really trying to capture. So we can label each residue within that sequence based upon the type of situational environment that exists within the protein. And we can map this across larger family complexes. And so rather than doing a sequence alignment, we can essentially do a sort of a structure-based alignment to look at not just is this sequence residue conserved, but perhaps is this environmental residue conserved? And so we've done this within a much more simplistic statistical approach, so hidden Markov models, to essentially capture how we think these proteins can be better represented. And so for each Scott, Cath, and ECOD protein family, we've generated an individual hidden Markov model based upon the 3DI characters that so our protein structural language characters that we've developed. And now we're using this to identify what we term as twilight enzyme activities. So we can take an existing protein family, generate our hidden Markov model, and we can identify these twilight activities a lot more accurately than what we could do from a sequence-based approach, primarily because structure is more highly conserved over a longer evolutionary time than sequence. Now, one of the issues that we came into with one of our industry partners when we started applying this to some of their problems is that you don't always have an entire protein family to start with. Sometimes you might have, we've got this single enzyme that we know works how we want it to, but can we find other enzymes that might have a very similar functionality? And so we've applied a slightly different approach for that, where what we do is we take that single protein structure, we perform molecular dynamic simulations on it. So we let it sort of relax over time, adopt different conformations if it does. And then rather than doing an evolutionary alignment to generate our hidden Markov models, we essentially do an alignment across those molecular dynamic simulations. And what's been really interesting to see is that the models that we're developing using these MD simulations have been just as accurate, or in some cases more accurate, than those being developed across entire SCOP or Cath protein families. And so this is one approach that we're looking at tying into the broader aims of the hub to really help filter down the types of proteins that we want to explore that might have similar activities to the ones that we're really interested in, but have more amenable protein features for further engineering. The other approach we've been using is more traditional AI approaches. And so they have really relied quite strongly upon our graph-based representation concepts. And so as an example, about 10 years ago now, we published a model called the PKCSM model that captured information from both a substrate perspective, but also the protein three-dimensional structure perspective to try and predict different pharmacokinetic and ADMET properties. But one of the aspects of that we were looking at was, could we identify which substrates would be metabolized by or acted upon by which cytochrome P450s, and could we do that in the reverse way as well and take a certain cytochrome P450 and identify all the potential substrates that it might interact with. At the time, we were limited a little bit by the data we had available, but also the types of models we were playing with. And so it was very much a classification approach at that time, where we were really just looking for yes, nos, and taking the probability of our prediction to rank them. But more recently, so just this year, we released an updated model of this called DeepPK that uses deep learning and a much more sophisticated data set that we've had access to through our industry collaborators to generate regression-based models. And while we're not quite at the level of accuracy that we would like to ultimately be, particularly as we link in with the data that Liz and Gary are generating in particular, we're really looking at hopefully refining these to be even more predictive than they are now. The other thing that we tried to do a few years ago now was, rather than just looking at a single enzyme activity level or activity function, could we predict all potential enzyme functions at the same time? And so we developed a multi-classification model called LEGO CSM that did exactly that. So it could take the structure of a protein or the predicted structure of a protein, and using a very simplistic machine learning approach with our graph-based signatures, it could accurately predict the enzyme class level, so the EC level down to class 3, and we still had over 60% MCCs at class 4, which was really encouraging, but also things like Go terms and subcellular localization. And so we've been using a combination of these two approaches to really explore novel and twilight enzyme activities to try and find new proteins that might be more amenable to biocatalysis and industrial processes. And so one example of those that we published a few years ago was in collaboration with a group at the University of Melbourne. And so they had an industry partner that was really interested in trying to find novel enzymes that could be involved or help with the methylation of mercury, so as part of a decontamination process. Unfortunately, the conditions in which they were looking at trying to perform these reactions were fairly high salt levels, and a lot of the terrestrial-based enzymes that they had identified weren't appropriate for their industrial processes. Our academic collaborators at the University of Melbourne applied a metagenomic screening approach to try and identify a subset of bacteria that may house these types of proteins. And the approach they used was to look at different taxonomic profiles across the depths of the ocean. So there's a particular area of the ocean, across most oceans anyway, that's enriched in mercury levels. And so we really focused in on sequences that we could identify from bacteria in those high mercury level regions. And so using our AI approaches, our structure-based AI approaches, we identified a couple of enzymes that we theorized based upon the structure, but not the sequence, would be had the potential to methylate mercury. And these were expressed, purified, crystallized, and then ultimately shown in the lab that they could methylate mercury under the conditions the industry collaborator was looking for, which was a really exciting proof of principle for us. But this isn't always the first step. In this case, we were really lucky that the enzyme we identified worked in the conditions we wanted it to, but oftentimes you still need to do a level of protein engineering beyond that to either stabilize the protein, make it more active, or change the levels of activities. And so one of the things that my group has in particular been really well known for has been the development of AI approaches to understand how changes in the protein sequence affect a broad range of functionality. So whether it's the stability of the protein, its solubility or expression levels, its activity levels or interactions with other molecules. And so where we first started this sort of push towards looking at mutational effects was really looking at the effects of mutations on protein stability. And that was really because that was where a lot of academic research was focused at the time, and there was a good set of data for us to play with, experimental data. But what we really noticed was that a lot of the methods that had been developed, even though they had reasonable levels of predictive performance across destabilizing mutations, they worked quite poorly, or I would even go as far to say were not predictive for stabilizing mutations. And this was in part a reflection of the data that was used to build them. Most academics are going to publish, I introduced this mutation and it destabilized my protein. And you get a lot more of those than you tend to get, oh, I introduced this mutation and it really stabilized my protein. And so we had to apply a few clever approaches to try and address this problem. And so this was really the work of a PhD student in the group, Carlos Rodriguez. And so we did two things. One was we weren't happy with the concept of just using a static protein structure. Proteins are highly flexible and dynamic. And again, we're losing information. So we integrated that appreciation of the potential movements that a protein could make into our algorithms. We also had to deal with the imbalance in the data, and we did that through the introduction of what we termed as hypothetical reverse mutations. The nature of the way that these things are tested experimentally is that you experimentally test the stability of one protein and then another. And so whether you're looking at the change from A to B or B to A, they should essentially just be the inverse of each other. And so we could use that to essentially create a perfectly normal distribution of our data. And when we applied this to independent test sets, what we could see is that our methods were just as accurate as at being able to identify and predict stabilizing mutations as they were at predicting destabilizing mutations. And we've applied this to a broad range of other concepts. So I talked about solubility and expression levels. We've applied it to looking at the effects on how proteins interact with other proteins, other nucleic acids, other small molecules, even metal ions. And so we've got a suite of about 20 or 30 programs in this area now where we've consistently tried to make improvements. And I'm happy to share further details about those offline if people are interested. And so what's been really encouraging to see is that the approach that we sort of initially pioneered and pushed forward in the protein stability space has really applied quite well when we've been looking at these other program developments, where the accuracy that we're getting across the stabilizing mutations, so increase in stability, is just about equivalent to what we're seeing with the mutations that decrease the binding affinity. So we're quite happy with this because it means that our methods are going to be much more useful and effective from a protein engineering perspective, rather than just trying to capture pathogenicity type approaches, which is where a lot of methods have really been focused on. More recently, however, we have really started to push into new approaches to try and deal with these issues. And so in work that's been pioneered by another PhD student, Emily, who's just finishing up at the moment, we've been looking at how we can apply different deep learning architectures to try and help deal with this imbalance problem. And so you may be familiar with the concept that a lot of deep learning algorithms rely upon the generation of something called a loss function. And so you essentially use the loss function to determine what's the difference in the predictions that we're getting between the predictions and what we would expect to see in the real world. And so Emily developed a contrastive loss function that was really designed to capture what was happening between these stabilizing and these destabilizing mutations. And so what we could essentially do is say, if we look at our stabilizing mutations and those reverse hypothetical destabilizing mutations, if the algorithm is working the way we expect it to, you should essentially have a zero loss function. They should be equivalent to each other. And so using this, we could actually then start to get away from essentially artificially manipulating our data and just using the real data that we had from the experiments. And so what you can see here is that, quite encouragingly, the level of performance that we see at the for the destabilizing mutations, which in this particular example are the mutations with a DDG less than 0, is very, very similar to the distribution of data or predictions that we get for stabilizing mutations, which is above 0 here. So the distribution, so the error from that regression line is about equivalent, which means that we're performing quite equivalently on our stabilizing and destabilizing mutations. Now, one of the things that I'm very passionate about is that I really don't like when people refer to AI algorithms as a black box. To me, an AI algorithm should give you some insight into the data that you originally used to build it, and also into how it's making that prediction itself. So all of our methods, we've always really focused on interpretable machine learning algorithms. And we did a very similar thing for this deep learning algorithm. And what we could see is that the features that we were using, or that the deep learning algorithm was using to really drive the prediction of these predictions, would align very closely with what we would expect to see just experimentally, or from a very experimentalist structural biologist perspective. So things like the hydrophobic contacts and things like that, which we know anecdotally and just from looking at protein structures play a really key role in how a protein folds and its stability. They came through just naturally in the deep learning algorithms as things that played an important role in the prediction. And so that was really reassuring for us that the way in which the algorithms were performing aligned very closely with the way in which we would expect that they should be making those decisions. And so we've applied this across a few different enzyme families to date. A lot of the data on those is commercially sensitive, and I'm not allowed to present it. But one of the examples that we did do that I can present was on a human protein called HGD, and it catalyzes this opening up of a benzene ring in the homogenate. And so our industry collaborator was really interested in ways in which we could try to optimize the thermal stability of this enzyme, because it's, like a lot of enzymes, metastable. So its TM was around about 36 degrees Celsius, which plays an important role biologically because it helps regulate the levels of the protein in the body. But from a functional and biocatalytic perspective, it's not particularly useful. And so what we applied here was what we termed in silico saturation mutagenesis. We mutated every residue in the protein to every other possible residue, along with all the permutations and combinations of those, which led to orders of magnitudes of possible protein sequences that we ran back through our programs. And what we were really looking for were mutations that could stabilize the protein. So in our predictive outputs, they would lead to a change in the free energy of stability of at least 1 kilocal per mole, and or improve the stability of the overall enzyme complex. So this is a homohexamer, so it's six independent chains of the same protein. And so could we stabilize that overall complex? But we didn't want it to reduce the binding affinity for the substrate, because that would ultimately then lead to decreased activity. So using our ligand prediction tools, we wanted to make sure that it would not affect the way in which the substrate could bind to the protein, or if anything, improve the binding of the substrate to the protein. And so from that, we came up with a list of 10 mutant sequences that we were really interested in. We expressed and purified all of them. I think it's the first experiment I've done quite like this, where I've expressed every single mutant that I've designed has actually expressed and purified and was soluble. It was quite nice. Hopefully the experimentalists in the audience are nodding, because it's not that common. And when we went to then measure the thermal stability, what we could see is that the introduction of just 2 point mutations led to an increase in the thermal stability of nearly 20 degrees Celsius, and it led to an increase in the expression of about 30-fold. So we're starting to get into production levels that we can actually start using and playing with. And I don't show it here, but the enzyme activity did slightly improve, but that wasn't the overall goal of the project anyway. It was just a nice side effect. We've also been applying this to a range of other concepts. So here you can see the alignment between the experimental measurements and the predicted data that we generate for looking at how an antibody binds to its target. And so even though you can see that there's fairly large error bars across here, the ability to rank our mutants across this broad spectrum aligns quite closely with the experimental data. And so we worked with a company in the UK to try and engineer a tick-borne encephalitis virus antibody. So they had this antibody that worked very well for one particular strain of tick-borne encephalitis virus, the Siberian strain, but it bound weaker to the European strain. And so what we did was we applied a very similar concept where we did in silico saturation mutagenesis across the entire antibody structure to look things that would improve antibody stability, optimize binding to the European strain. And we generated a panel of 6 mutants that we could then go off and test. So our industry collaborator tested them. And of the 4 that expressed, so in this case, 2 didn't express, all 4 did improve the binding to the European strain. And one in particular, they were very excited by it because it improved quite significantly the binding to the Siberian and the European strain. And in this particular example, we then went on and ran a second round of in silico mutagenesis where we identified a second mutation that was quite complementary to that initial tyrosine 32 to methionine mutation, and that improved the binding to both the Siberian and the European strain even more significantly. But I'm not allowed to show you the double mutant, just the single mutant. And so over the last 10 years, my group's developed over 70 different tools across this space to really try and help improve rational protein design, our understanding of how proteins function. And these have been implemented and used in a broad range of industry collaborative projects. They form the foundation of a number of academic platforms. So as an example, the PDBE uses the way in which we calculate and map interactions as part of their visualization tools. And so all of the software that we've developed is open source and freely available, and you can access it at this link at the top. If you have any questions or want to use any of it, we're more than open to collaborate or to help point you in the right direction. So please feel free to reach out. But now I'll hand over to Lisette to tell you a little bit about how this will then integrate within the broader business concept.
Yeah, thanks, David. And I just want to reiterate your welcome message from earlier. So it's great to see you all online today and looking forward to having a discussion about biocatalysis. So one of the other approaches in this hub that we're really interested in exploring is how can we streamline decision-making, looking at the differences and the benefits of biocatalysis versus chemocatalysis and how it can be implemented to create value within a production system. So one of the overall aims that we're going to be addressing is how we can build a data-informed system to guide that decision-making process from the very beginning of any development of new process procedures, so that the decisions that we're making are guided by data from the start of the project, but also the start of any of the production systems and processes. And so we're going to be looking at different costs and yields from different production systems, applying, generating models to understand those costs and predict those costs, and identifying the really important and really important business variables that are important in that process, such as the existing infrastructure, personnel and resources, any of the economic outlook and returns that are important. So the first component will be doing business analytics and economic modelling of the bioreactor formats and enzyme production systems, and how can we maximise value of those systems, and what kind of information and data do we need to run models to understand value maximisation. The next approach is to think about techno-economic and life cycle analyses of these processes to see if we can understand and determine optimal combinations in terms of chemo and biocatalytic steps, using models that have been developed within the University of Queensland here, and then also bringing in some AI and machine learning algorithms to develop decision trees and start to predict production systems that might be more efficient to maximise value. And this is drawing off my background, and my contribution to this project comes from my background in data analytics. As Liz mentioned, I'm really interested in understanding efficiencies and innovation speed. I've done a lot of research in this space, looking at efficiencies and dynamics in clinical development in the pharmaceutical sector, and also the impact of novel technologies and how that's impacting innovation speed and efficiencies through the clinical development process. So one piece of work that we've done that's really looked at how clinical trial efficiencies have been evolving within the pharmaceutical sector is looking at changes in clinical trial length. So we have a data set of over 100 or just under, 100,000 clinical trials that were registered at clinicaltrials.gov, which is the leading clinical trials registry in the US. And based on all that trial data, we were interested in looking at how has the length of those clinical trials changed over time as sponsors have developed experience and built capabilities in running and managing clinical trials. In order to do that, we had to build a regression model so that we could control for all of the factors that we were able to measure that could impact trial length, such as the phase of the trial, the type of indication, how the trial was designed, and the experience of the sponsor, to measure the average length of clinical trials over time and see if that had changed, increased or decreased over the period that we were interested in. So we found quite clearly that the average length of clinical trials across all phases had been decreasing, and also been decreasing depending on the type of sponsor who's been sponsoring the clinical trial. We found industry-sponsored trials were actually already running more efficiently and quickly than trials sponsored by other types of organizations, particularly universities, other research institutes and government-funded research institutes, which, although not super surprising, was still interesting to see reflected in the data. And that research was published in Nature Reviews Drug Discovery. Since then, we've been focusing more on broader levels of total clinical development length across different pharmaceutical products and the impact of regulation and how regulation can improve the speed and efficiency of the clinical development process at the new drug level. They've done some work looking at the impact of a new regulatory program that the FDA introduced in 2012 called the Breakthrough Therapy Designation. Many of you aren't familiar with this designation. It's designed for sponsors to work almost collaboratively with the FDA to improve the speed and efficiency of the clinical development process for drugs that are showing initial really strong clinical data and which address an unmet need in the market. And the overall aim is then to get these new drugs to market as quickly and efficiently as possible to help benefit patients who need these new novel medicines. We found that the impact of this breakthrough therapy designation really did have a benefit on the length of the clinical development process, but also in how the clinical trials were designed, which we linked back to the efficiency of the system. So we were interested in looking at different clinical trial designs in terms of surrogate endpoints or use of endpoints, how they were controlled, blinding and randomization. If there are differences in these trial design characteristics based on drugs that had received the breakthrough therapy designation to assist their development process or didn't, but also when in their process, in their development history, did they receive that designation? And so did they receive the designation before the trial had started versus after the trial had started? And how did that affect the design of the trial and then the overall length of the new product development process? I've also been interested in new technology development in the pharmaceutical sector, particularly around precision medicines. So precision medicines are new drugs that have been developed or targeted at patients based on their genetic or genomic characteristics. And we identified, based on a data set of nearly 150 or 180, I think, novel drugs that were approved by the US FDA over a period of about 5 years, and how long it took those drugs to advance through their total clinical development period. We found that the medicines that weren't classified as precision medicines took around 7.5 years to advance through their clinical development phase. And this was defined as the time period between when the sponsor submitted the investigational new drug application to the FDA and when they when that NDA or then BLA was approved and the drug was approved or licensed for marketing. Compared to 7.5 years for medicines that were considered non-precision medicines, so not targeted based on genetic or genomic characteristics of the patient or the patient's disease-causing organisms in this case as well, versus drugs that were considered precision medicines advanced through in under 6 years, so nearly a 1.5 year saving in time, which is quite significant when we're considering the total length of pharmaceutical new product development. We found further increases in efficiency and speed to market for the precision medicines that also received that breakthrough therapy designation that I mentioned earlier. Those drugs came through their clinical development process in under 5 years. So that's an extraordinarily fast time frame for clinical trial speed or trial length for drugs in the pharmaceutical industry, as I'm sure you're all aware. And created a nice benchmark. This work was actually done before COVID. Created a really interesting benchmark as to how quickly new technologies were getting through their clinical development process with the assistance or almost collaboration with the regulator through this breakthrough therapy designation. But I also have experience in qualitative research, particularly using qualitative research methods to do deep dives to understand problems at a really deep level, as well as thinking about the context within the industry that firms are operating in, to understand what are barriers and enablers to efficient product development and commercialization. One piece of work that we completed recently was looking at the impact of new regulation in digital health technologies, particularly in the Australian market, and how could digital health technology developers engage with the regulatory process early to help provide or help improve the efficiency of their new product development process, so they weren't having to go back and repeat development steps to meet the regulatory requirements. And this is some work that we then continued during COVID, looking at how regulation and the regulatory process in general could help or hinder development of novel vaccines and how that could be applied into the COVID situation. So back at the very beginning of COVID, we actually started some work around about February 2020 in this space. So right when COVID was becoming, or apparently becoming a potential problem, was to look at how quickly could we actually get a COVID vaccine to market. There's obviously reports of at the beginning of COVID that it was going to take around 12 months, which if you recall, based on the precision medicine data we previously analysed, seemed very, very quick, considering the precision medicines, even with new technology and with the help of the FDA, was still taking nearly 5 years to get to market. So to understand how we could maybe get a COVID vaccine to market in under 12 months, we had a look at the preclinical history of 6 different vaccines that were developed in response to prior epidemics or pandemics, and how quickly they proceeded through their preclinical development and then were able to start clinical trials. We found that with the exception of a flu vaccine in response to one of the influenza pandemics in 2009, none of the other vaccines that were being developed in response to epidemics or pandemics over the past 20 years, such as Ebola, MERS, the first SARS, Zika virus, actually completed their preclinical development experiments and stages and started clinical trials and finished clinical trials and were then approved on the market in time to impact immunity with the pandemic as it was progressing. The only vaccine from the pandemics and epidemics that we investigated that actually made it to market was Ebola vaccine, and that was 5 years after they started development and long after the Ebola epidemic was over. So what were the bottlenecks that prohibited those vaccines from getting to market as quickly and efficiently as possible to have an actual impact on the immunity of the epidemic or pandemic? Thankfully, we did see COVID vaccines come into market within 12 months. And so some subsequent work was looking back and reflecting on what were the new regulatory pathways and processes that were put into place to enable that to happen. And what were all the different ways that well, in this case, we looked at the FDA as a case study. How did they put these practices into place to enable and assist the vaccine developers in advancing those vaccines to market quickly and efficiently? And what can we learn from that to apply to either future pandemics or even going beyond into our I wouldn't say standard, but non-pandemic vaccine development processes. Some work that I've moved on to more recently, particularly with the assistance of one of my doctoral students, is to look at this idea of business models and how firms can develop new business models to improve their efficiencies and also deliver more value in trying to develop new products for the pharmaceutical industry. So this is coming from the perspective of business models as an interlinked system of all the different activities that's performed by a particular firm. So all the steps and processes involved in turning inputs into the firm into outputs to capture, deliver, and create value, including all the networks and relationships that the focal firm has with its suppliers and customers. So what's all the different activities in this system that a focal firm performs to create, deliver, and capture value? And taking that one step further and looking at, well, how can that business model change or evolve to improve value capture, delivery, and creation activities, create more value, and create and sustain a competitive advantage in the industry. And this idea of business model innovation has become really important in recent years as a complement to traditional product or process innovation for firms to conduct to create and sustain a competitive advantage. So realizing that developing new products is one part of a way firms can create value and sustain a competitive advantage, but also how are those products delivered to the market? So the work that my doctoral student completed was specifically looking at the microalgae industry, in that what are some, I think his research question, well, what were new business models that could be implemented immediately within this microalgae industry to improve the value of the industry, improve the sector in general, and create more value? Given the current policy and regulation requirements, the current level of technology development, and the current market dynamics. It's one thing to say, well, what could we do in the future? But what can we do right now to create value, given the current technology that we're working with, the current market dynamics, and the current policy situation? So in order to meet that broader aim, Alvaro identified all the different core drivers of actors participating in the microalgae space, both in Australia, and he also looked at a couple of overseas jurisdictions. And based on that identification of core drivers, he designed novel activity systems that was rooted and building on that evidence base, and then looking at how those new activity systems can create new business models that can contribute to sectoral development and basically improve the value of the sector. And this was achieved through exploratory, qualitative, and cross-sectional research, specifically semi-structured interviews. So he looked really specifically at the motivations, internal constraints, external constraints, the needs, as well as the value creation, capture, and delivery goals of organizations in the microalgae sector and opportunities that were identified. And I was able to identify multiple novel activity systems or business models that could be implemented immediately within the current technological requirement and framework and policy environment to create value in this industry. So this is an area that we would like to explore further in this research hub, in that we could take a structured approach to business model mapping across biocatalysis and chemocatalysis processes. So taking this idea of business model innovation and applying it at the process level to understand the barriers and enablers of implementing biocatalysis into existing technology domains using the current market dynamics. So how can we create value immediately using biocatalysis into existing production systems? And if we can take a couple of pre-existing but also recently developed business model mapping and product development feasibility tools to bring a structured approach to our analysis. So one approach that is gaining a lot of popularity in recent years is the business model canvas, and its sister, the value proposition canvas. And these are great tools developed out of research, I believe from Stanford University, to enable organizations to map their business models by looking at what are the key activities that they perform, what is their value proposition, what are the revenue streams, cost structures, partnerships. So by looking through this canvas, we can make sure we have a structured approach to understanding activity systems, as well as how we can create value with those activity systems through alleviating pains in this case in businesses, but in our case in the chemocatalysis process, and how can new biocatalysis processes alleviate those pains to create new value gains. So that's a great model that's come out of, as I said, I believe Stanford University, but also complementing this structured approach by using models that have come, one particular model that's been developed here at the University of Queensland to look at feasibility of implementing a new process within an organisation. We call this the commercialisation tourbillon, because by moving through a series of 12 steps to understand market dynamics and competitor dynamics, for example, and financial opportunities within a particular process, we can become closer and closer and almost spiral in to a value proposition for how new technology can be implemented into an existing system. And so just, sorry, just the tail end of a cold. So just bringing all of this together and bringing some of the work that David's been doing together, in this hub, we're planning on leveraging our collective backgrounds and expertise to undertake business analytics, as well as economic modelling to look at the value of different bioreactor formats and enzyme production systems, so that we can really maximise value and efficiency in the production processes for the pharmaceutical industry. So how we plan on actually operationalising that is, first of all, to take some of those core business model structured tools that I mentioned earlier to map the industry activity systems and identify the drivers or core drivers of value in both chemocatalysis and biocatalysis systems through in-depth qualitative research and interviews, which we always conduct under proper ethics approval and requirements. In Australia, that's driven by the National Health and Medical Research Council. And we can combine those in-depth interviews with desktop research and market research to identify trends across the industry and how we can maximise value, but also answer specific questions that are posed by our industry partners. So an example of this has already come up in these webinars in the past, and through discussion with potential industry partners. We were, I guess, alerted to a potential opportunity in that how can we use the existing equipment that's used in biocatalysis and modify that for existing equipment in the chemocatalysis process, and how can we modify that to use in a biocatalysis process so that we're not having to, I guess, invest in expensive new infrastructure. And so this approach, and by creating new developing novel business models and activity systems, we would be able to answer that question. The next step is to take this in-depth, nuanced and contextualized understanding of barriers and drivers and enablers, and taking this structured approach with the business model canvas to develop regression-based techno and economic models. Using our previous research in the first aim, or the first part of the project, to identify all the different dependent, independent and control variables that we need to include in the economic models. Sorry, it's Friday afternoon here. We've been speaking for a lot today. Our economic models, so that we can start to measure efficiencies from both an economic and a technological perspective of chemocatalysis versus biocatalysis, generate data for those variables from experimentation across our other projects in this hub, and then use that model to measure different bioreactor formats and enzyme production systems and their corresponding value. Taking that one step further, we would also then like to bring in some more artificial intelligence and machine learning to take this model and try and make it into more of a predictive model, training it using the current experimental data, but then how we can use that to predict bioreactor formats and different enzyme production systems that can maximize value in the production process. And then use these AI predictions to then further accelerate and direct the experiments that we're doing to really accelerate this, the research that we're completing in this hub. So overall, we feel like these two areas of expertise that David and I bring to the team really help us to combine deep learning through artificial intelligence with the techno and economic analysis to really understand how we can create more value through these different production processes, which ones would create the most value, or alternative forms of value, and how that can, I guess, ultimately impact the bottom line of our industry partners. So thank you for your time today. I'll hand back over to Liz, and thank you for your time.
Thanks very much, Lisette and David. And I will close off the recording part of this webinar because we don't want to record the question and answer sessions in case there's anything that people don't want recorded. So the recording will cease now. But can we ask if there's any questions from the audience here?